Thank you, Danny, for the presentation. So I'm gonna talk to you about how part of Korf research in Macaronaceae is related to my research during my PhD and my postdoc. And so all of you know about probably what is an inoperculate disco. Korf was a great specialist in Discomycetes. And he influenced a lot my job during the first stage when I was doing my PhD. And one of the things that I have to point out about inoperculate discos is they are really small, but it's a really interesting group. Leotiomycetes and Orbilliomycetes, you can find them worldwide in any ecosystem that you think. And they have a really great variety of lifestyle. So that makes this group of fungi really interesting to me, to learn about their ecology, because we really don't know much about them. So I'm from the Canary Island. I'm from the island Tenerife, the one that you can see here in the middle of the archipelago. And I did my PhD there in the University of La Laguna. And the Canary Island is known very well in Europe because we have a really good climate. So we have mild temperatures through the year, and the season that is rainy is usually between the end of autumn during winter and first part of spring. And we are influenced by trade winds, so we have we can call like a subtropical climate. But sometimes we have sandstorms that come from Africa and make the weather more hot and dry. Something really beautiful that you can see when you go to an island like Tenerife that has a lot of altitude, almost 4,000 meters. It's like from the coast to the summit, they are very differentiated ecosystem with very different type of vegetation, with each of this type of vegetation have their own microclimatic condition. During my PhD, I was studying the Leotiomycetes and the Orbilliomycetes in all of this type of vegetation that you can see here in the pictures. So before I start my PhD, I was reviewing what was reported to the Canary Island. It was around 80 species reported between Leotiomycetes and Orbilliomycetes. And here you can see how they were distributed in the old concept of the family, of course. So most of the reports were in Yellowstone. Probably you know the genus Lachnum. A lot of reports were also in the Medici. I imagine that all of you also know Moyisia. And then you have here some pictures about other easy genera that we can identify in this group of fungi, like Ascocorani, Orbillia, Leotia, or Rusraimia. So in this volume of Mycotaxon, you can find a beautiful text that talk about Korf and me. And Korf was working in the Canary Island. During the period of time of my PhD supervisor, she was starting being a professor. So he visited the Canary Islands several times, and my PhD supervisor, Esperanza Beltran, she was working with him like 3-4 times they go together to the field. And in this volume of Mycotaxon, you can see that talk about how Korf helped me during my PhD. He was even close to his death, helping people like me. We are going to talk a little bit about this. So he published 15 works about Discomycetes, and 10 of them are about inoperculate Discomycetes. We cannot go through all of them, so I choose 4 genera that I think are really interesting, and I can talk to you about all of them because we are going to find really interesting thing in each of these genera. But before I start to talk about that, I would like to remark that what are the difference between how Korf was working in the lab and how I'm been working. So I will say that Korf is like old school, like he was going to the field, collecting, drying the specimen, and study them months or years after. He or his student studied most of this collection when they were dead. So basically all the cells of the fungi that you are seeing are dead, and you rehydrate them in water or KOH, and then you study the morphology. On the other hand, I've been very influenced by Baral's method. So when I started my PhD, I was not only in contact with Korf, but also with Baral. And it was really fun to be in contact with both and have discussion with both of them. And I worked mostly the micromorphology using Baral's method, what we call vital taxonomy. And vital taxonomy is really important in this group of fungi, and because it gives you easy morphological characters that you can use to quickly identify families or genera or even species. So the main thing with vital taxonomy is you must know how to differentiate living and dead cells. The meaning of this is like, if you go to the field and you collect something, maybe it's not alive. Sometimes you can have collections from the field that are 1 year old or more, and they're dead. But usually when you collect something in the field and it's fresh, it's living. But you have to be careful, and you must know how to differentiate living and dead cells. And of course, today I have the advantage that Korf didn't have, to use DNA, and also like a tool that helped me to compare what I'm finding also with my morphological studies. So the important things that you can see here, for example, when you are studying living cells, and you can see gutules in the excipulum, that you cannot see them when they're dead. The cross symbol, it means dead, and the asterisk, it means alive. So in the Orbilliomycete, for example, you cannot see the spore body, which is a structure that is fundamental if you want to identify Orbilliomycetes. In the paraphysis, again, you cannot see gutules. And something that is also important is when you are doing vital taxonomy, you must study the specimen fresh when they are alive. And then you usually also kill the cells using reagents, so you can compare the differences in measures, because there are differences also in measures. So it's very important that you know that you have to be very careful with the reagents. If you use KOH, you are going to kill the cells. If you use Mercer, you are going to kill the cells. So they are going to be smaller, and for example, the arrangement of the spore inside the assay is not going to be the same. You are not going to be gutules inside, so depending on the nature of the gutules. But you can use some reagents like Congo red or crazy blue, which can give you some contrast and also help you to see better the walls of the cells. So taking that in consideration, we're going to start with the genus Mollicia. I'm guessing that all of you know the genus Mollicia. So I love the title of this publication, made by Greenleaf and Korf in 1980, Mollicia in Macaronaceae, an exercise in frustration, of course. This is still my frustration too. And in the moment that Korf and Greenleaf were studying this genus, the genus Mollicia has a broad morphological concept. So basically all flat disco that grow on wood and have globose dark cells could be a Mollicia. And then there were other genera like Tapetia that have subiculum, or Aglundia that have hairs, that after they were erected, they were synonymized again in Mollicia. The interesting thing about this publication, of course, they did a lot of the effort. You can see the drawings that they did about putting really clear the characteristics of the excipulum. But if they study probably more than 100 collections, they only were able to name one species. They erected Mollicia dextrinospora, and the name is because the spores turn red when you put it in iodine. But all of the other collection were naming using letters. So you have Moyisia A, B, C, D, and all the letters of the abecedary. Even they were at the end another Moyisia that was A. So unfortunately, during my PhD, I had the same experience. So I contacted Korf. He sent me his Moyisia with these names with letters. And after sampling for 2 years, I got like 121 collections, and I was unsure about the identity of most of them. Even if I was using beetle taxonomy, and we have some keys in Europe from Baral and Andreas Suminder, I was not able to be sure about the identity of most of the species. And I saw them in 19 morphotypes, and maybe someday I'm gonna be able to go through again, but right now they are in a folder in my laptop, waiting for maybe to have the opportunity to start to work on them again with DNA. But the interesting thing is like the only species that they publish with a name, Moyisia Spinospora, well, it's not even a Moyisia, unfortunately. Some people using DNA placed it in the genus Cadophora like years ago, but I'm right now collaborating with Brown, and I review the type in CUP, and I also got several fresh collections from the Canary Island, and so we are thinking that the correct genus for Moyisia Strinospora is Pyrenopesiza, and we have been reviewing different species in Pyrenopesiza because we believe that really exists an older name. Right now, we have the idea that maybe Pyrenopesiza aquosa, it could be the same that Moyisia Strinospora. So other of Korf research in Macaronesia, it was about the genus Orbillia. And he reported 8 different species in the genus Orbillia. And in the moment that he was working in the genus Orbillia, Orbillia was considered a low diverse genus placed in Leotiomycete, which is really interesting because then when Baral started working Orbillia with vital taxonomy, show that Orbillia is really diverse. And I imagine that maybe all of you know the monograph that was recently published. So when I start my PhD, I was finding Orbillia everywhere. And at the end of the PhD, we realized that we had 63 species. Several of them were new to science, and even some of them are really endemic and very host specific. But how is possible that Korf only found 8 species and I found all of these species, all of this diversity? Well, this is because most of the researchers that come to the Canary Island, they usually go to sampling in the same ecosystem. They usually go to mid-elevation, which are the most humid ecosystem, like pine forest, laurel forest, or plantation of castanea and eucalyptus. Most of the report of Inoperculate discos are from these hosts. Most trees in the laurel forest or on Pinus canariensis or on castanea or eucalyptus. But because of my sampling design during my PhD goes from the coast to the summit, I was working also in the same ecosystem, but also in 2 new ecosystems. In the coast, in the Euphorbia scrub, which is a really interesting vegetation with a lot of succulent euphorbia that can look like cacti. And also in the summit brune scrub, which is very interesting ecosystem also because you have important differences between the day and night temperature. Usually it doesn't rain, we have a snow. And in the Euphorbia scrub, during a whole year can very rain 1 or 2 times. So what I found that Korf was not able to find is that most of the species of Orbillia are in the Euphorbia scrub. And you can see that in the laurel, in this box plot, that is representing the average of the number of species that I found in Orbillium in this different type of ecosystem. You can see that the Euphorbia scrub, the number is higher than the other three ecosystems. And it's really interesting because you realize when you represent the host plant diversity in the Canary Island, the Euphorbia scrub is the ecosystem that have more in the missing, talking about plants. And you can see that the distribution, that the diversity of Orbilliaria is following exactly the same host plant diversity curve. When we go on to check about the composition of a species, we found two different groups. So in the humid ecosystem, in the laurel forest, and in the pine forest that is humid, usually mixed with the laurel forest, the composition of a species is very similar. And the same happened when you compare the dry ecosystem, the summit brown scrub, the Euphorbia scrub, and the dry pine forest. And if you check here, each of these lines is like the altitudinal range of each species that I found. And you can see that a lot of these species are in the coast, in the Euphorbia scrub, and they don't go higher. And then we have some species that can go from the coast to the summit. And that's the reason, because I found a lot of more species than Korf, because I was studying this dry ecosystem. Here you can see like different species. This is like a simple analysis that basically takes the abundance of the species and explain you what are the more important species by vegetation. And you can see how the first species is different in each ecosystem. And in the laurel forest, for example, we have Orbillia santostigma and yellow Orbillia lunata, which are not in any the other ecosystem. The third example that I would like to show you here is about the Geocoraini. I don't know if maybe some of you know this genus. It was selected by Korf. And he elected this genus taking two different species, one from Micronesia, Geocoraini varispora, and another one from India, Geocoraini exogloea. Geocoraini is very interesting because it's one of the few, I would say, Leotiomycete genera that grow on soil, usually grows on clay soil among mosses. And in that moment, when Korf erected this genus, he included in the family Leotiaceae and say that the closest relative were Ascocoraini and Pesoloma. So when I was doing my PhD, during the first years, I never found a fungi like this. And then I was collaborating with the Mycological Society of Tenerife, Gran Canaria, and also people from Madrid, from the Mycological Society in Madrid that come to the Canary Island. And then we start to find this fungi that you can see here that looks a lot like Geocoraini. But the interesting thing, there were variation in color and in size. And we, of course, before we work the morphology, and then we go through keys. And then when we go through some keys, we always reach the genus Dicinella. And Dicinella was selected by Boudier in 1885. So in this moment, you can go through Ascofran and see there are a lot of discussions between Baral, Korf, and Miguel Angel Rivas about I've been collecting this in the Canary Island. I think it's Geocoraini, and then Baral is discussing about this is not really Geocoraini, it's probably Dicinella. So there were a lot of discussion during my PhD, so I decided to work a little bit about this, and we are still working on this. So I finished my PhD in 2015, so we are still working on this. And here you have a drawing of Geocoraini varispora in the left, and you can see the gel layer in the xipulum, which is a character very interesting, that go from the base to the margin. And you can see exactly the same in the pictures in the right. These are collections that we have from the Canary Island, different places, and what we think is what Korf called Geocoraini varispora. I'm saying what we think is because I'm taking in consideration the type of study that I did. So we have fresh collection that agree with the type. But Korf include in Geocorynebarispora the type and other collections. So what really happened here is that when Korf erected Geocorynebarispora, he didn't realize that indeed he was collecting Dicinella, and he was collecting different Dicinella. So the pitted Varispora, the variability he found on spore size and morphology is because of that, because he was mixing collection of different species of Dicinella. And here you have a phylogenetic tree that is showing a little bit things that we have sequenced. Now we have more sequence, but while still working on this, because it's not very easy to get collection of the different species in the genus Dicinella. And you can see that we have sequence from the type, and we have a clay that we have here the name Dicinella boudieri. You can see there are a collection from Canada. So maybe this species is widespread more than we think. We know that there are some collection calling Geocorynebarispora from Ecuador. But then you can see that in the Canary Island, we have also Dicinella boudieri, and we have something that we believe is Dicinella menziesi. And this is still a project that we are doing because there have been several problems. And one of the problems it was like I request the types from PC for Dicinella boudieri and Dicinella menziesi, and they were lost when they sent the type to us. So right now we're still struggling with that and also trying to get more collection from different parts of the world. Now we have also collection from the South Hemisphere for Dicinella boudieri and for other species. So unfortunately, Korf never realized about that Dicinella was published before, and this is one of these interesting cases that a person that knows a lot about this group of fungi didn't realize about the publication of this genus and erected a new genus. And to finish my talk, we are going to talk about Clausenomyces. So Korf also worked together with our lead Clausenomyces in Macaronesia, and they published 3 species like new. 2 of them were collected in the Laurel Forest. One of them was collected only in the Canary Island in the pine forest. You have here the drawings that he did for these 3 species, Clausenomyces clavatus, Dacrymycetoideus, and canariensis. So when I was doing field work, I was finding Clausenomyces constantly, from the coast to the summit, in each ecosystem, in different host plants. Clausenomyces is very common in the Canary Island. It's really very common. And the problem that I was having was that I cannot really name all of these collections that I was finding in the names that Korf published, because, for example, I have 2 different Clausenomyces that look like Dacrymycetoideus. I have like 5 different Clausenomyces that were looking like clavatus. I have 3 different Clausenomyces that look like canariensis, and I was realizing about differences because of the use of vital taxonomy, and also because I was using different features that Korf didn't use. And then I have other things that I was calling Clausenomyces that were none of these 3 species that Korf report to the Canary Island. So because of that, we were finding this interesting diversity in the genus Clausenomyces, and there were not much information. And at the end of my PhD, I started working on this, and then at that moment, Baral was working in the syllabus of Leotiomyces, and he was interested that because I was doing that, why you don't also compare, he said, with genera that we believe are similar, like Tympanis, Durandiella, Drachmopora. So at the end, the last part of my PhD was mostly a revision of the family Tympanidaceae, a quick revision, not really a revision. So we got some collections, and I tried to get sequence for some of these genera. We put everything together. We did a first phylogeny with ITS and LSU, and we found that some of them were related. And we also find that we didn't have really good support for the family. But at that point, Baral decided that, the morphological evidence is good enough to, right now, let's put all of this together in a new family, and we will see what happens in the future. And I agree with him. And so the family Tympanidaceae was published in 2015 when I was finishing my PhD. And then instead of publishing this chapter of my PhD, I changed it in a project. And I started my postdoc here in Harvard University. Something that is really interesting in Clausenomyces is that you have a species, what you can go through the literature and find these two words, primary spores and secondary spores. So what they are, this mean that you can find like normal spore, like usually 8 spore in the outside, like the one that you can see here in the left. But then sometimes these primary spores can produce secondary spores. The way that we differentiate this secondary spore is using 2 different names. So when we have an aside that have spores that produce conidia from the wall, like you can see here in the picture here, and usually the aside is dead and you can find it like a few conidia attached to the spore wall. We call them conidia. This secondary spore, we call them conidia. But then there's something really interesting, and some species of Clausenomyces, what they have is like these primary spores are surrounded by like a membrane. And these spores inside the membrane start to produce a lot of this secondary spore. And at the end, what you can see in the assay is like a ball with a lot of conidia. So when this happens, like the figure that you have here, each of these conidia ball that we is ejected like a unit, like it wasn't a spore, but in these hundreds of conidia. So when this happens, we call them as coconidia. So we differentiate. We don't have conidia, we can have conidia, or we can have ascoconidia. And you can only see this feature of ascoconidia usually when you are doing beetle taxonomy, when the specimens are fresh. So in Clausenomyces, you can find different species that have these different features. So during my postdoc here, we improved the taxa sampling, and then I'm showing you here some examples that I presented in one congress. Right now, we know that what Korf called Clausenomyces, like 2 years ago, I think we combined in the genus Vexillomyces. So most of the species that you know of Clausenomyces are now naming like Vexillomyces, and are those that have ascoconidia, these balls filled with conidia. We proposed also some years ago that Clausenomyces prasinulus should be called Dendrostilbella prasinula because of the anamorph. We proposed to conserve this name instead to propose a new name for this Clausenomyces that we know is not really a Clausenomyces. And then we have some species like Clausenomyces olivaceus that grow on resin, very different substrate, is related to some of these Tympanidaceae like Tympanis and Durantiella. But the thing is, like, probably none of them are really related to the species. So this is the type of species, Clausenomyces jackianus. Bjorn was talking about rare fungi. So I've been years and years and years trying to get a fresh collection of Clausenomyces jackianus, talking with people all around the world. And finally, a year ago, somebody in Czech Republic collected. So now we have beetle taxonomy for Clausenomyces jackianus, which is the type of species of Clausenomyces, and you can see it's very, very different. So it's the only Clausenomyces that is a rumpen through bark, and is big compared with all the Clausenomyces, Apothecia. And the asci are really long, and the spore are as long as the asci. You can see the spore here. This is the type study, but when we found this fresh collection in Czech Republic, it was sent to Adam Polhorsky in Slovakia, and he did beetle taxonomy is the same that the type. And now we have cultures and we are trying to get different genes to put in a phylogeny and show what we are mostly sure that the type of species is not related to anything that we are calling Clausinomyces. So right now I'm also collaborating with Baral and other people, you know, like putting all of this information together and try to do like an overview about Clausinomyces, what is really Clausinomyces and everything that we have been calling Clausinomyces. Thank you for your attention. And I would like to thank the support that I've been having from different money sources and also the people that collaborate with me, from researchers, you know, amateur mycologists, and staff in the Herbarium or librarians. And if you have any questions, you can always email me and ask me whatever you need.
Hey, Luis, I have two remarks, no questions.
Okay.
You can congratulate you for your nice talk and even for in the end pointing it out that you have shown a prime example that it's absolute nonsense to use the terms sexual and asexual morph because morphs that can produce conidia. Huh? And the second thing, I also like it because I'm now struggling with sort of for a long time that we should culture these things. And you also have provided a good path forward in the end. We have to get cultures, we have to get multi-gene genealogies to make our things watertight. And I really believe this will be great to achieve more clarity about this complicated fungi in the future. So I really like your work, Christine. Huh? I really can say it's really very great what you presented to us today. Huh?
It's very difficult to understand with the environmental sound. So I don't know if you are.
Might be.
Yeah, can you repeat it, Danny? Very close to the mic.
I can come back.
Yeah.
So to cut it short, the first thing I like this that you have pointed out a very good example with the Clausinomyces, you know, with the stuff that the ascospores can already form conidia.
Yeah.
It is nonsense to use the expressions asexual morph and sexual morph. This is really a good thing. And the other thing is I like that you are starting to culture your things. And I think if you had cultured your specimens when you did your PhD, you would already be more advanced, huh?
Yeah.
Yeah. I can only encourage everybody to culture their things, at least if they grow.
Yeah.
It's also mean to of vital taxonomy, huh?
Yeah.
Okay.
Yeah. You
But
always
you really did a super good job, Luis.
Thank you. Yeah.
And maybe
yeah,
I come back to Tenerife in December. Maybe.
Oh, I will be there.
Okay.
Yeah. Perfect. Yeah, I always say that you start a project, but you never know when you are going to finish it.
Yeah.
I have a question, Luis.
Okay.
Let's say which Discomycetes broadly, or which Leotiomycetes more specifically, benefit the least from vital taxonomy, and in essence are the most similar, whether the tissue is living or dead?
Well, for example, in Tympanidaceae, it's not that bad. You know, I can work a living tympanis and dry tympanis collection that are really old, usually are very abundant, which is something really good. And there are not really many differences. I mean, you have differences in the size, but at the end, that's not really important because there are not features like in Orbillia, you know, that you need. There are not these features like the spore bodies or gutules that you cannot see, because most gutules on Tympanidaceae are lipidic bodies, so you can see them, you know, in Herbaria collection. So Tympanidaceae is a good example that you can work everything dry, it's going to be fine. But for example, it will be very difficult
Wow.
In general, the thing is, it's very easy when you are working with vital taxonomy to differentiate, for example, genera like Bisporella and Calycina. You cannot differentiate them from Herbaria collection. You cannot differentiate a true Bisporella, from Calycina. Most of people are collecting are Calycina, not Bisporella. Even if they call him Bisporella, probably most of them are Calycina.
I remember.
And you have a lot of examples like that. It's like for example, when you are working in Leotiomycete, you can differentiate sometimes Sciaticola and Hymenocipus and other genera because of the backwards in the paraphysis. The pattern and the number is really informative. In Moysia, you can also have something really interesting. Is like when you work in them fresh and then you add KOH, you have reactions. Some of Moysia never release pigment, others release pigment in different colors. And this is really important to identify them.
That was a kind of a follow-up question. With Cordillera-Tidacea, does would a nionomydotic reaction be different with fresh material or dry material?
We can discuss a lot about the iadomydotic reaction because it's so unclear to me. I mean, I've been having this discussion with Don, a lot of people about what we call a iadomydotic reaction. For example, Tympanidaceae should have iadomydotic reaction. Most of them, Korf said that you can find iadomydotic reaction in Clausinomyces. I never find it. So I think we have to differentiate the reaction in iadomydotis, what happens when you add KOH to some species, and what happens sometimes is like you have what I do is like I mount the section in water, and I let them dry a little, and then I add KOH, so I can see how the KOH is going inside the tissue, and then I take several pictures. And usually what you see, for example, in Clausinomyces is like the tissue expands and the pigment change through the tissue, but they are not really releasing of pigment, like you can see in iadomydotis. So I don't know, it's kind of the iadomydotic reaction. I always have this problem, trying to understand really what we are calling iadomydotic reaction.
It's actually like with our survey, since we know which compounds are responsible, we can interpret these things better.
Right.
So we have to take this I think Louis has really shown, really demonstrated very nicely that we can now move this thing to another dimension.
Yeah, exactly.
You hear that? You have to move it to another dimension.
I will try to.
And I have a lot of trouble with Soto now because he insists that IPS should be the holy grail. You will not believe it. Soto is my good friend, but you can argue with him forever.
Yeah, that's true. But it's been better with years.
Yeah.
After years and years, it's been better. I hope I'm going to meet him in person in the next Mycological Congress. In the next international. No, no, it's amazing. I've been working with him like 15 years and we're still not met in person.
He will hopefully come
in Amsterdam.
Now, I hope you will all be able to come, or you should consider, because next year we are arranging this, and this will be a nice conference. It will be worthwhile to come to Europe. We can even move sort of Barra out of his sofa. Hopefully.
That's not getting me.
No, so that doesn't go to the field anymore for some years because he tells me he has too much things to do. He really fears if he goes out and collects more, that he will never get finished. And I'm so happy that he finished this beautiful monograph after 27 years. You cannot imagine.
Yeah, I think it's an amazing book to learn. Go through the monograph if you want to learn about vital taxonomy, and it's so well explained in everything. In morphology, before it was published, of course, I was using part of the monograph during my PhD, to interpret the morphology, to describe. It was really, really helpful. And I think it's really beautiful work. Huge. And since it was published, I've not been using it so much. So I hope I'm gonna use it at some moment again.
Okay. Thank you, Luis.
Welcome.
Oh, and what do we need to find to bribe you to the next Korf Foray?
I hope so. Yeah, it was really unfortunate that I'm leaving to the Canary Island in 1 week, so I was really not able to go.
But we have to have some bounty species, some things that if we find, then you cannot help yourself and you have to be here next time.
Okay.
You'll let us know.
Louis, if you come
Yeah.
back, say hello to me for me to Esperanza.
Yeah, okay.
So last
Yeah,
time we couldn't meet, we had some around, but we come back in December.
Okay, perfect.
Yeah.
Okay. All right. Thank you again, Luis.
Bye. Good night.
Go on.